Good morning, everyone, and welcome to the last day, that is sixth day of 1 week International Faculty Development Program on Herbal Medicine Research and Global Health, a therapeutic perspective, organized between 9 to 14 Feb by Other Shikshan Prasarak Mandal, KT Patil College of Pharmacy, Usmanabad. Ladies and gentlemen, education goes beyond shaping us as individuals and enhancing our perspectives. It can help us to become a better human and build a better society. With this refreshing quote, let's begin our new day with a new perspective. Dear participant, pharmaceutical industry is constantly searching the ways to ensure and enhance product safety, quality and efficacy. However, the drug recalls, manufacturing failure costs, scale-up issues and regulatory burden in recent past produce huge challenge for industry. In traditional, the product quality and performance are predominantly ensured by end product testing with limited understanding of the process and critical process parameters. Regulatory bodies are therefore focusing on implementing quality by design, a science-based approach that improves process understanding by reducing process variation and enabling process control strategies. Recently, the concept of quality by design gaining much attention among pharmaceutical industries for maintaining quality. It serves as a bridge between industry and drug regulatory authorities to move towards a scientific, risk-based, holistic and proactive approach for development of pharmaceutical products. It mainly covers designing and developing formulation and manufacturing process to ensure predefined product quality. The purpose of today's session is to discuss the concept of pharmaceutical quality by design and how it can be helped to ensure pharmaceutical quality and drug development. So, for to know more about quality by design, we have a very eminent personality with all of us. I would like to give my heartiest welcome to the today's resource person, Dr. Jayaprakash Sangshetty sir, Associate Professor, YV Chavan College of Pharmacy, Aurangabad. We are so fortunate that we have got an expert personality like you, sir, in our panel today. And thank you so much. You have accepted our invitation within short request and ready to give a talk in our program. Before we start our session, I would like to introduce our today's resource person, Dr. Sangshetty sir in brief. Dr. Jayaprakash Sangshetty sir is presently working as an associate professor at YB Chavan College of Pharmacy, Aurangabad. He has completed his B Pharm from YB Chavan College of Pharmacy, Aurangabad. He has completed M Pharm from KM Kundanani College of Pharmacy, Mumbai, and doctoral degree from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, under the guidance of Dr. Devanand Shinde, former Vice Chancellor, Shivaji University, Kolhapur. He has around 19 years of professional experience. He is approved PhD guide of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, and 6 students are working under him. He has 4 patents, 3 are published and 1 granted on his credit, and has published 235 research papers in international journals with a cumulative impact factor more than 400 in Thomson Reuters 2020 and H index of 34. He authored 4 books. He has written 2 book chapters in books published by Nature, Springer, and Bentham. His research work has received 3,610 citations. He is reviewer of more than 70 international journals. 5 of his research papers have been ranked by Science Direct as the hottest 25 articles. He is a recipient of Young Scientist Award from DST, that is Department of Science and Technology. He has successfully completed project received from DST worth rupees 25.52 lakhs, from UGC worth rupees 6.2 lakh and AICT worth rupees 13.8 lakh. One extramural research project from SERB, that is Science and Engineering Research Board, worth rupees 36.55 lakh is in progress. He has received a Best Paper Award from Elsewhere in 2015. He also has received Young Pharmacy Teacher Award from Maharashtra State APTI in 2015. He has also received the Life Best Publication Award in 2014. He has received PD SETI Award for Best Research Paper in 2009. He has guided more than 35 M-Pharmacy students. He is presently working as a member board of studies of pharmacy at Shivaji University, Kolhapur. He is working as an international panel member of Project Evaluation Committee of National Science Center, Poland since 2015 and evaluated member of projects. He is also working as a brand ambassador for Bentham Science Publisher since 2017. His major area of research include design and synthesis of bioactive molecules, development and validation of analytical methods for drugs and pharmaceutical dosage form, application of quality by design in drug development, synthesis of polymers and their application in gene delivery system, Recently, he has been included in top 2% researchers list published by researchers of Stanford University for the year 2019 and 2020 consecutively. An academician, an innovator, and a very renowned and experienced speaker. What to tell about the achievement that they have got in their career in short time of period. We have a indeed privilege to have you, sir, in our panel today, sir. It's my honor to welcome the resource person of today's session, Dr. Jeff Sanchez, sir, to our distinguished panel and would like to request to start their presentation. Please.
So, very good morning to one and all. So, am I audible?
Yes, sir. Yes, you are
Yes,
audible.
yes. Okay. So, thank you, madam, for my introduction. So, I'm here to discuss one of the important aspect of pharmaceutical regulation, that is quality by design in pharmaceuticals, with special emphasis on uh, uh, analytical method development, so which is an integral part of all sectors of the pharmaceutical development. So, analytical development or developing the analytical methodologies, it is, as I mentioned just now, is an integral part of drug development. So we all are in a, we can say, era where industry or industrial requirements or regulatory body requirements are in a transformation phase. So what type of transformation is going on? What industry will we can featured for, and what will be the future of pharmaceutical industry as far as drug development process is concerned? Being an academicians, we should be always in touch with all these particular current trends of drug development and in regulatory requirements. So the very basic objective of this particular lecture is to overview what quality by design is and how this quality by design concept can be we can say applied for analytical method development. As such, no specific guidelines are available, but with the available guidelines and the expected guidelines, which we are expecting within 1 year through the ICH guidelines. So, based on the concept paper which has been published by the ICH, what parameters we need to consider while developing any suitable analytical method, that we will discuss. So, giving an overview of quality by design with a special emphasis on analytical method development, that is the objective of this particular lecture. Now, as we know, pharmaceutical development, it is a process which is directed towards one common motto, that is quality products. So we all are working, all pharmaceutical sector, it is working with the product where the quality of this pharmaceutical product is that our prime goal. And considering this requirement, all the activities are driven with this common objective. Now, rather than going in the basic definition of quality, so what we are looking for the quality products, and regulatory bodies are more concerned with the quality of the pharmaceutical product, and with that requirement, number of changes, number of amendments in their regulatory requirements, regulatory bodies are continuously doing. Now, what current approach we are following as far as quality products are concerned, so that we are calling it as an quality by testing. So in the current approach, we are testing the quality of the pharmaceutical product at the end. And after the testing, we are taking the decision about that entire batch which we are evaluating. Again, large number of lacunas, large number of limitations are there with this particular approach, which we are calling it as a quality by testing. Now, as such, whenever a new science enters in, we can say, in the field of research or in the any sector of science, so that particular science or that particular concept must be having some advantages against the existing concept. And that is the idea, where whenever something new comes as a part of regulation in the pharma sector, then it must overcome the limitations of the existing concept. And with that view, since this particular approach which we are following nowadays, this approach we are calling it as a quality by testing, it is having certain limitations. So to overcome these limitations, this quality by design approach has been accepted. And how, what makes regulatory bodies to accept this particular quality by design approach, that we'll discuss in coming slides. Now, what quality by design is, to summarize this slide, quality by design, it is a science-based approach which is totally based on understanding the process. So there must be some theory behind it, and that particular theory that we are discussing in the coming slide. So it is a systematic approach to the pharmaceutical development that defines with the predefined objective. So we have to define the objective at the start. We have to work in order to achieve that objective rather than testing the product at the end. We have to develop the product with a desired quality, that is the quality by design. So a systematic approach which is based on the science, it is having a sound support of science, and it is totally depending on the quality risk management. So this is the definition of quality by design which has been given in ICHQ 8. Now, as such, this particular tool, that is quality by design, which is not at all a new science that we have invented, these pharmacy people have invented. This particular quality by design tool, it is well established in the engineering sector since last 50 years, more than 50 years also. So chemical engineering people, they are using this particular quality by design tool for large number of developments of the processes. So what is the basic of this process? The basic of the process is this tool or this particular science, it is totally depending on understanding the process. So when we are working on any process, then understanding the process is very important if you want to identify what critical parameters are there in that particular process. So quality by design, it is totally depending on the simple tagline is studying that particular process, going in the depth of that particular process, identifying the important parameters that are responsible for or that are contributing towards the success of that particular process, controlling all these particular parameters or steps which are very critical, and implementing the continuous improvements tool. That is the most important aspect of quality by design. So quality by design, it is a science which we have accepted from the engineering science because this particular tool has been well established in the engineering field, as I mentioned just now. Now, what is the basic difference between the current approach and quality by design approach? So we are following the traditional approach where we are judging the quality of the product by testing and inspection, where quality by design, it is totally based on the building the quality or developing the product of desired quality. So current approach, it is a data intensive submission, whereas for QBD approach, it is a knowledge rich submission. Now, in case of current approach, specifications are based on the history, whereas in case of QBD, specifications based on the product performance requirement. So current approach, it is a frozen approach where there is a less chance once you submit any application to the regulatory body for the approval. But in case of QBD, we can change the process even if you submit any application for any regulatory body. So it is comparatively flexible approach compared with the current approach that we are following. Now, what is the main focus of the current approach? The main focus of current approach, it is on the reproducibility. Whereas the main focus of this quality by design approach, it is only on the robustness of the method, understanding of the method, and controlling the parameters or variations of that particular method. So it is an empirical development in case of current approach, whereas systematic development, which is based on the science, which is based on the quality risk assessment. So these are some of the important points that can be considered as a differentiating points between the current approach and quality by design approach. Now again, as I mentioned, this particular approach, it is based on a thought which has been given by a quality expert, Joseph Giron. So rather than testing the quality, the product, you can develop a product of a desired quality. So that is the thought which he has given. And on this thought, different engineering teams are working in the product development. And we have adopted this particular thought to address large number of regulatory requirements, large number of regulatory issues that we were facing at the start of 2000. Basically, as I mentioned, whenever any science or any concept we are accepting, then that concept we are accepting to overcome something which we are facing. Now, this quality by design, it has been accepted by the regulatory bodies to address number of issues. And what were these issues that we'll see in the history of this particular quality by design approach, why we have adopted this tool, why regulatory body feel that this tool needs to be adopted. So basically, in 2000, uh, regulatory bodies, they were facing tremendous pressure because of evaluation of number of files. The reason behind submission of number of files was not the files which were submitted at that time. They were not for the new applications, but they were actually the supplements. Supplements were submitted usually uh, uh, with reference to the initial submission that has been done previously 1 year back or 6 months back. Why the supplements were there? Because they initially they have applied for some pharmaceutical process, and the inventor or the pharmaceutical manufacturer want to make certain changes in that particular application. So, so large number of supplements have been submitted, and which were more than double than the original applications that have been submitted. If the 2,000 applications were submitted, more than 5,000 supplements were submitted by the pharmaceutical manufacturers. And evaluating all these supplements was a major issue in front of regulatory bodies. So for evaluating 5,000 supplements, the time required was much more. And they thought that all the regulatory bodies, which were the key regulatory bodies at that time, they thought that we need to bring all the pharmaceutical manufacturers on a common platform. We need to give some common guidelines in order to submit the different applications with reference to NDAs and ANDAs. And with that thought, uh, regulatory bodies sat together and they have decided to give some guidelines in order to apply for NDAs and ANDAs. And that is the start where QBD approach has, we can say, uh, entered in the pharma sector. Now, again, in order to implement quality by design, they have not directly implemented quality by design as such, but they started because they want to change the entire system, and changing the entire system, submission system or application system at a glance, it is very difficult, and that's why they have initially started the first step, and that first step is CGMP. In 2002, FDA implemented changes, some of the changes of submissions and different guidelines as far as manufacturing is concerned through CGMP. And this CGMP, it is the first step for implementation of quality by design. Now, later on, some of the advanced guidelines have been provided through this process analytical technology, which is again a system for designing, analyzing, and controlling manufacturing process, which is based on again understanding the process, understanding the science and factors which affect the quality of the final product. In 2005, they have taken the first step as such as far as implementing QBD is concerned. So they have asked some of the firms or some of the manufacturers to submit their applications in the format of we can say question-based review, which is known as QBR. And this QBR, it was the first step which has been taken by FDA, US FDA specifically, to implement QBDs to some sections of industries. Later on, the guidelines of QBD have been included in ICH, and ICHQ 8, 9, and 10, they have been, we can say, framed, and they have been approved, and these guidelines were available, made available in 2008-9. And initial guidelines were available. In 2013 and 14, US FDA has made this quality by design mandatory for generic products. And from that point, they are slowly accelerating the drive to implement this quality by design concept. Means implementing a new science, we can say, or a science for a particular concept in pharma development sector. As such, it has been started in 2002, and from 2013-14, actual implementation of QBDs for generics, they have been started. Then again, in 2013-14, the questions were raised about what about the API? And since there were no specific guidelines for API synthesis as far as quality by design is concerned, then in ICH guidelines, different guidelines have been framed, concept notes have been published, and later on, they have given the guidelines in different ICH guideline forms, that is 10, 11, 12. 13. Up to 13 ICH guidelines have been published and ICH guidelines are available. Now, if you see this particular historical perspective of this quality by design, it is clear that coming era, that is the era which is where the pharmaceutical sector it is more focusing on, it is quality by design era. So every aspect of pharmaceutical drug development, starting from, you can say, the API development to uh, to formulation development, all these sectors will be under the quality by design format. And that's why it is our concern that we should be thorough with the concepts which are there and how this quality by design can be implemented to the different sections of drug development. Now, again, as I mentioned just now, when initially the concept has been, we can say, adopted in 2005, and after 2005, 6, 7 implementation of quality by design through ICH guidelines, QBD has been considered as the combination of three ICH guidelines, 8, 9, and 10. So in 2011-12, again, people raised the questions about what about API drug synthesis. Then in ICHQ 11, some guidelines have been given related with the drug substance. So drug substance means API manufacturing and the, how this API manufacturing can be processed so that the number of supplements associated with the API, new API synthesis will be reduced with the regulatory aspects. Then later on, product life cycle management, it is the concept again which is based on quality by design. ICH has given the guidelines in ICH Q2, which is where the overall, if you want to develop any product and if you want to make any changes as far as development process is concerned, all these changes will not be considered as a change if the change is not significant. And for that, the concept of, we can say, design space has been given, which is a key part of this quality by design. And product life cycle management, ICH guidelines, they are again based on this QBD. Then continuous manufacturing of the drug substance and drug products. This is again a part of quality by design, and these particular guidelines have been implemented in 2021, where the concept paper of the same has been published in 2018. Now, the next part which we are expecting from the regulatory body is what about the analytical method development? Analytical method development, it is an integral part of the pharmaceutical development. We need the different analytical methods. We need to analyze the different starting materials, APIs, or intermediates involved in the API synthesis, along with the APIs, excipients involved in the formulation, along with the finished product. So use of different analytical methods as per the requirement, it is always there as far as drug development process is concerned. Now, how we can say perform this analytical method development in the QBD framework, and people are again continuously inquiring with the different regulatory bodies about any guidelines regarding this use of we can say analytical method under QBD framework. Then in November 2018, ICH has given the concept paper before implementing any ICH guideline. So they are publishing a concept paper. Concept paper, it will give some picture, you can say, not 100% clear picture, what type of guidelines will be there, but it will give some guideline, what will be the content of that particular ICH guideline after the draft. And in November 2018, a concept paper has been given by ICH, and that particular concept paper, it was related with analytical method development, which involves some of the guidelines which are related with the quality by design for analytical method development, and some revisions of ICH Q2, which is again analytical method development and validation. So why these two things have been combined here? Because some of the guidelines have been already given by ICH in Q2 related to analytical method development and validation in 2006. So these guidelines have not been, we can say, amended, or these guidelines have not been improved. Why there is a need to improve that particular guidelines? Because these guidelines have been specifically related with some of the equipments, specific like HPLC. But current, if you see the current scenario of drug development or new drug discovery, there are many analytical tools like near IR spectroscopy, which has gained much attention in last 1 decade. The protocols or the guidelines related to the development of analytical methods using this near IR technology or near IR spectroscopy, it has not been mentioned in ICHQ2. So ICHQ2 specifically speaks about the analytical method development, and more focus has been given on the routine analytical tools like UV and HPLC. So, as I mentioned just now, near IR spectroscopy, it is widely used as far as new drug discovery is concerned. And no specific guidelines have been given in ICHQ2, and these guidelines needs to be updated. And that's why in ICHQ14, along with some new guidelines related with the quality by design analytical method development, advancements of the previous guidelines, that is from Q2, will be there. And this concept paper has given some information about this Q4. Now, what will be the future of analytical method development as under QBD roof, we can say? So that has been already discussed in the concept paper, and it is based on some of the previous literature. And that particular literature I am going to discuss here. What should be the content of ICHQ 14 as far as analytical method development is concerned? Now, again, this is a concept paper which I just now mentioned. So it involves two sections. The first section, it is related to the some of the guidelines related to analytical method development under QBD framework, and some of the guidelines which are related to the advancements or which are related to the some of the analytical tools which were not there in ICHQ2. For example, near IR, Raman, NMR, and MAS, because hyphenated techniques like LCMS or LCMS-MS, they have been not included in ICHQ2 because at that time these analytical tools were not much explored as far as drug development or drug discovery is concerned. So advancements or some of the amendments in the existing ICHQ2 along with the some guidelines related with the analytical method development under QBD format, they will be included in this ICHQ14, and these guidelines or the draft of these guidelines is expected to publish by the end of December. Now, what parameters? If you go through this entire concept paper, this concept paper is of 4 pages. So I have summarized some of the important points in this particular presentation as far as different guidelines are concerned. So this is in general what QBD is. So before understanding the basic requirements for analytical method development, we should know what exactly quality by design is. So quality by design, as I mentioned, it is totally depending on uh, uh, achieving the task. That task has been predefined and working on the entire process. For working on the process, we need to understood the process. If we understood the process, we are getting an idea about what are the important aspects that we need to focus. And once you gain an idea about what are the critical aspects of that particular process, then we have to implement the control strategy. And once you implement the control strategy, the errors in any process, this is in general about quality by design. If you speak about any of the specific process in the pharmaceutical development, then we have to define the objective, what you want. If you want to talk about any analytical method development, what you want to measure, to what extent you want to measure, what sensitivity you are expecting from that particular analytical method. If suppose we are talking about any of the pharmaceutical development process, if we are talking about a tableting, what type of tablets you are expecting, what hardness you are expecting. So such objectives we need to define at the start, and accordingly we have to work. We have to identify what are the parameters that are directly contributing to achieve that particular objective, and all such parameters are always considered as an critical parameters or critical quality attributes. Now, identifying these particular critical parameters, assessing the risk involved, then minimizing the risk involved by using design experts and developing the experimental design. And once you gain the control on that particular method, okay, then how we can gain the control? If you control the risky parameters involved in any drug development process, ultimately we gain the control of the process. once you develop the method, during their actual usage, if we are facing some problem, then there must be some scope for continuous improvement. In previous case, we don't have the scope for continuous improvement because that particular approach which we talk about, that is quality by testing approach, it was a frozen approach where once you submit any application to the regulatory body, in that case we cannot make the changes. If you want to make the changes again, we have to submit that applications in the form of supplement. And in case of quality by design, we need not we can say go for any additional supplement, because we are giving we can say in the application only we are giving such information where any change that we made in that particular design space cannot be considered as a change as far as quality by design is concerned. Now, again, different analytical tools where quality by design concept has been successfully implemented through the literature. We can find large number of hyphenated tools also. They have been worked out with the under the we can say using different primary guidelines which are available as far as quality by design is concerned. Now, what are the advantages? As we know, in initial discussion, I told that this quality by design, it is totally focusing on the robustness parameter. Robustness of any method, which is again an important validation parameter. So the method which we develop using or adopting quality by design, it is more robust compared with the routine method. Then this particular method gives a greater transfer success, because developing the method it is quite easy, but transferring the method from different levels of the development is quite difficult. So people are facing the problems as far as technology transfer is concerned. So if particular product can be developed at a we can say R&D scale, then transferring that particular technology to the pilot scale and then to the plant scale face number of challenges, and this particular number of challenges can be addressed to major extent, we can say, using quality by design. So this particular tool provides a space for invention of new techniques by continuous improvement. Continuous improvement is one of the important aspects where we can make the changes during development. Even if we have developed certain methodology, and if you find that this particular parameter needs to be improved further, then we can improve that particular parameter, or we can implement that particular change. And this is possible only in the QD framework and not with the routine, we can say, quality by testing framework. Now, as I mentioned, in case of concept paper, number of aspects that they are going to consider as far as new guidelines are concerned. But the people who are involved in framing these ICH guidelines, they have initially published some of the data. If you see the body who is involved in framing these guidelines related to analytical method development in QBD format, then these are the researchers who are working in the different pharma companies, and they have worked on the different analytical tools. Now, large number of efforts have been taken by the researchers to fit this particular analytical method development in the QBD format, and they have given the different approaches. So whatever the guidelines will become, these guidelines will be based on the two important reports that has been published by the renowned research groups from the different sectors. So the first approach, it is MDS approach, which has been given by Wolf et al. In 2011. Now he is again one of the member who is involved in framing the ICH guidelines, and probably what he has published at the in 2011, some of the guidelines may reflect with his particular thoughts. But if you see the literature, overall, we cannot change the we can say a framework of quality by design. Again, as I mentioned, quality by design, it is a risk-based approach. It is based on the understanding of process. So minimizing the risk mitigation we can call, and gaining the control on that particular process. These all are the important strategies. So every aspect which I am explaining as far as analytical method development is concerned, it is applicable for both these aspects. Now, what he has suggested. According to this particular group, this MDS approach, which has been published in 2011, so they have initially decided what method goal is, then what they want to measure, to what extent they want to measure. So impact of analysis on the product and safety, then intended use of the method and end user, then
ऑनलाइन chat post में टाका पड़े.
Hello, sir. Excuse me, sir.
hello.
Sir, your PPTs are not visible, sir. Can you check it, please?
Okay. Hello.
Yes, sir.
Yeah, madam, I've given the PP PowerPoint presentation to Umre sir also. He can
Yes,
share from
yes,
it.
I will check it,
Okay.
sir. Yes. Just 2 minutes, sir. We are presenting our slide.
Hello.
Yes, sir. Just a
हाँ
minute,
मैडम.
sir. Yes, sir. We are presenting, sir.
यहाँ से एक और एक बार ट्राई करता हूँ मैडम मैं. जस्ट वेट. Okay.
Sir, which number of slide is it?
16.
Yes, sir. 16.
Okay. So as far as this particular approach is concerned, am I audible?
Yes, sir, yes. Yes, sir, yes.
Okay. Okay. So as I mentioned, this particular approach has been given by Oak et al. In 2011. So this was a particular research group from GSK, and they are actually involved in framing the ICH guidelines, that's Q14, which are expected this year. That's why what should be the actual picture of this ICH guidelines? Based on their initial research work, some of the important aspects they have suggested in this particular approach. Now, if you see this approach, so they have initially mentioned that initially we need to set the method goal. So while setting the method goal, what should be the impact of analysis on the product safety? To what way this particular analytical method it is helpful in defining the quality and safety of that particular product. Then intended use of the method and end user. Then based on the physicochemical information about the analyte, or the particular substance, it may be active pharma API, or we can say other excipients which are present in the drug product. So that particular information helps in setting the method goal. Once the method goal has been set, then method scouting and evaluation, which type of method is suitable for analysis, that will be decided in the method scouting and evaluation. And once the methodology or experimental tool has been decided, then actually when we enter in that particular method, then what should be the method conditions? We have to perform the risk assessment. Then you have to go for robustness studies. And once you go for the robustness studies, and once you evaluate what should be the risk in that particular method, then we have to decide what should be the control strategy. So again, as I mentioned, the important aspects in general quality by design concept, the same aspects have been considered by them, and hopefully this particular all parameters may be the part of ICHQ14 in future. Now, in after the initial efforts taken by this particular group, again there are some important aspects that they have mentioned in this MDS approach. So these particular aspects we have covered. So in case of method goal, what is to be measured and with what limit it is required to be measured, that will decide what should be the goal of that particular method. And this method goal provides the framework to method development, which helps in further planning. As far as method design is concerned, large number of parameters have been considered. Permutation and combination of number of parameters that affect the performance of the method is considered. So, and based on that, the design space is developed. So, design space for any analytical method, it is again a multi-dimensional combination of parameters that affect the method performance. So, all these parameters needs to be identified, and the combination of parameters will be tried in order to reduce the risk involved in that particular method. Then method design is prepared considering the availability of the material and different experimental conditions. Then different reagents required. Then feasibility of the instrument. All these parameters needs to be considered while giving the method design. Again, some of the important examples we have given. In case of HPLC, method development scouting is done. All these are the parameters needs to be considered when we actually when in 2011 it was just the start of implementation of the concept where people have started to implement quality by design in the pharmaceutical development section. Much guidelines were not available. People were working considering the basic concept of we can say quality by design in analytical section. And that's why these are some initial hints given by this particular group related to the chromatographic method development. And these particular initial hints have been helpful. And further, this work has been taken up by another group that I'm going to mention later on. Then, as far as the design space is concerned, again, it is a multi-dimensional combination of interaction of input variables and process parameters that have demonstrated to provide assurance of the quality. Now, again, as which has been defined in ICHQ 8, based on the initial design space that you have created. Okay, movements within the design space, it is never considered as a change as per the regulatory requirement. If you want to make certain changes, then these changes are not considered as a change. So based on the design space created, experimental designs of different types which we are using previously can be used in analytical method development also. Now again, CQAs, understanding the process in case of analytical method development and product whose analysis is to be carried out is very important. Since we are concerned with the quality, so any type of impurity, it is always considered as a critical quality attributes as far as analytical method development is concerned. The level of impurity, the detection limit of that particular impurity, along with the ability of that particular method to identify that impurity is always considered as a critical aspect as far as analytical method development is concerned. Then for risk assessment, different types of tools can be used, like Ishikawa fishbone diagram, FMEA, and Pareto analysis. Pareto analysis, it plays a significant role as far as analytical method development is concerned. It has been most widely used, whereas for other types of pharmaceutical developments, all three can be used. But in case of analytical method development, Pareto chart is most widely used. Now, once the risk is identified, okay, then the segregation of the risky parameters into high risk parameters and low risk parameters is very important, and that particular role is helpful for gaining the control strategy, which is the most important part. As far as analytical method development is concerned, control strategy is one of the important aspects. So once you gain the control strategy and once you implement that particular method, then again the question has been raised in around 2010. One of the research paper has been published. What about the method qualification? Whether that particular method qualified as we do in case of method validation parameters. Then method qualification, again, they have some of the researchers have included the important part of this analytical method development under QBD environment, and this particular method qualification has been divided into method installation qualification, method operational qualification, and performance qualification. Where operational and performance qualification, these are the very important parameters as far as method qualification is concerned, because method installation qualification, it is a part of the installer from where we have purchased the equipment. Now, these are some of the important parameters which needs to be checked as far as and how this particular method qualification and performance qualification can be performed using HPLC that has been discussed. Now, as far as control strategy is concerned, once you segregate the parameters, go to the next one slide, please. Next slide.
Yes, sir. Yes.
Next one. Again, come to slide number 32, where control strategy is one of the important step in. Yes, next one. As I mentioned, in case of method goal, what is to be measured and to what extent it is to be measured, it is very important. Then, as far as method design is concerned, next one, please. So, slide number 24.
Just a minute, sir. Is it okay, sir? This one?
Yes, it's 24. Okay.
Yes.
So, design space, as I just now mentioned, it is the multidimensional combination and interaction of input variables, that is material attributes, and the process parameters. So, success of any analytical method, it all depends on how effectively the method works. This analytical method works, and how we can say you have understood the material attributes, means properties of that material or analyte under consideration. So understanding all these properties, and we can say the understanding the process, the key steps involved in the process, the parameters involved in the process. So combined effect of which will help in deciding what should be the design space. Hello,
Yes, sir. Yes. Please continue, sir.
मैडम, यहाँ से अभी शेयर कर रहा हूँ मैं, क्या अभी
जी सर, जी सर, जी
जस्ट एक बार देख लीजिए शेयर
जी
हो रहा
सर।
है।
अभी ऐड नहीं हुआ सर, एक बार देखिए सर। जस्ट अ मिनट सर।
Okay.
Yes, sir. Now it is visible, sir.
Visible? Okay.
Yes, sir. Yes. Please share your slides, sir.
Okay.
Yes, sir.
Now,
Yes, sir.
as far as critical quality attributes are concerned, factors which directly affect the quality and safety of product, if you are concerned with the product development, if you are concerned with the analytical method development, the process. Now, as far as CQA is concerned, identify is always considered as one of the important CQA in analytical method development in the pharma sector, because these particular impurities, since we are concerned with the quality of the pharmaceutical product, so impurity always considered as CQA in all types of analytical method developments. Now, as far as risk assessment is concerned, these are the three tools which can be used for risk assessment. Out of these three tools, Pareto analysis is most widely used tool as far as analytical method development is concerned. Then once you identify the risk, segregation of the risky parameters needs to be done. So high risky parameters and low risky parameters needs to be separated, and the more focus is always given on the high risky parameters to plan the important part as far as analytical method development is concerned. Researchers, they have included a method qualification, that is method installation qualification, operational qualification, and performance qualification as the important part in the method development, out of which the last two, one, that is operational qualification and performance qualification, always been considered as an integral part of the development of analytical method development under UBD environment. Hello. Hello. नाही आले, यायला लागले आता ते मी शेअर केलं स्वतः. Now, these are some of the important parameters that can be considered as operational qualification and performance qualification for the HPLC instruments. Now, once you identify the risky parameters, then high risky parameters, we need to gain the control on the method. Gaining the control on the method, it is again depending on, say, uh, how effectively you have segregated the risky parameters, and how will you gain the control on all these particular risky parameters. That is very important. Now, as I mentioned, once you gain the control on the method, then validation, it remains a formality. So usually in case of quality by design framework, QBD, it is developed based on some initial method objectives, and we are exploiting, we can say, robustness parameter to a wider extent, and validation remains a formality. So it is just as per ICH Q2, validation is considered as a method objective. But in case of quality by design, validation remains a checkbox tool rather than a method intent. That is the most important aspect. Means, in case of quality by design, analytical method development, we did not go for the validation. So that was the thought which was been given in 2011 only. Now, this particular aspect has been further, we can say, simplified by another research group, that is uh, George Reed. Again, they are from the Pfizer. So a research group, they have given analytical workflow in different way. The terminologies they have used, they are synonymous to the terminologies. The different terms have been used, but the meaning of that particular terminology, it is same as in case of previous one. Now, if you see this particular workflow which has been given by the Reed et al. In 2013, they have published this particular article in initially, in case of method goal, they have given as an analytical target profile. So in analytical target profile, we have to define the requirements and performance criteria. And once you define the analytical target profile, we have to select that particular analytical tool. What you are expecting from the analytical method. Based on that, we have to go for selecting a suitable technique. Using suitable technique, you will develop analytical method. And once you develop the analytical method, you have to go for the risk assessment. And for reducing the risk, you are going for the DOEs, and you have to determine the edges of failure. And once you reduce the risk involved in the method, you are going for design space was there in earlier approach. And in this particular approach, it has been named as an method operable design region. So method operable design region is nothing but a design space which they have been given, and anything or any change that you want to do once the method is developed, that is not considered as a method operable design region in case of this particular workflow. And if you go thoroughly through this particular workflow, it is observed that once you set ATP, once you decide the ATP, once you choose the analytical method, once you develop the analytical method, then if suppose you want to make certain changes after the development, and if the change is within that particular MODR, then that particular change is not considered as a change, and we need not go for another supplement application to the regulatory body. So this is another analytical tool which has been given by Reed et al. In 2013. Now, what are the key features rather than going in much detail? So they have given, I have taken the example of HPLC, and they have considered this HPLC method development under QBD environment. Each step they have considered as a different wave. So while considering what QBD is exactly, QBD, it is totally depending on understanding the process. Now, if I want to develop an analytical method for any substance under consideration, then I should know what that substance is, what level of analysis I'm expecting for that particular substance, and what analytical tool I am going to use, and what I am expecting from that particular analytical tool. So the questions of answers of all these particular questions definitely help me to we can say design the strategy as far as analytical method development is concerned. So what they have done, they have divided this entire analytical method development protocol into different waves. Initial wave consists of information about the drug, where primary information about the pH and log D, or which is related with the pK of that particular drug. Then about the experimental conditions, stationary phase, which type stationary phase you are using, which mobile phases you are using, and in next case we have to that is wave 2, we have to go for optimization of the pH based on the pH. pH plays a significant role. pH of the mobile phase along with the pK of that particular analyte under consideration. They play a significant role. And based on the pK of the analyte under consideration, pH of mobile phase needs to be set. That will help in we can say designing properly, or we can say developing mobile phase properly as far as PVD framework is concerned. Then in third wave, temperature and gradient screen needs to be done, where again different experimental approaches have been tried. And once all these particular aspects have been considered, in the last stage, the drafting of all the conditions based on the initial screening of different method parameters that they have been done. So that was the approach which has been given by Reid et al. In 2013. And these two approaches will be considered some guideline as a guidelines in drafting all these ICHQ 14 guidelines which are expected. So summary of this Acuity workflow. This particular Acuity workflow which has been given by Reid et al. It starts with understanding the method needs, followed by technique selection and selection of initial chromatographic conditions. Then refinement of these conditions in wave 2 and 3, and risk assessment to identify, and DOEs to reduce the risk. Then end result is robust chromatographic method, and with well understood MOD or DR, which was design space in the previous case. So these two particular methodologies that have been we can say suggested by different research groups working in the renowned pharmaceutical firms. So these have a common, we can say, basic principle. Here, in first case, they are named as a design space for that particular region where we can make the changes. But in this case, they are named as a method operable design region, and we can make the changes in both cases. Now, how we can proceed if you want to develop analytical method? For that, I have taken one case study in order to understand. So these are some of the important literature reports how we can develop analytical method in QBD environment. Now, there are no fixed or written documents or guidelines which are available. By the time these guidelines will come, we have to rely on these two approaches which have been developed by the different renowned research groups, and people are working on these guidelines. Now, suppose if you want to work on any analytical method development of any chemical, it may be excipient, it may be a drug, then how we can proceed in order to understand that? I have taken one case study, and this particular in-house case study we can say. Now, again, HPLC method development by QBD approach, a case study which will give some guidelines. We cannot say that this particular work which we have carried out, they will be the final guidelines as far as working in analytical method development using QBD approach is concerned. Now, if you see the different steps involved, literature survey and problem selection, identifying the drugs, HPLC method development by QBD approach. So based on the information which we get by the two approaches that have been suggested by the different researchers, we have initially defined the analytical target. Then we have gone for the instrument qualifications, risk assessment, and determination of critical quality attributes. So again, these are some of the initial attempts that we have made as far as method development is concerned, as I mentioned just now. So they may not be the final, we can say, guidelines as work. Then design of experiment to de-risk or to reduce the risk involved in the method development. And lastly, validation, which is again not a method intent, but it is a checkbox tool, which I have just now explained. Means validation is not mandatory under QBD environment, but we can perform the validation, which will just a checkbox tool whether the method developed is validated or not. So it will give an additional assurance about the developed method. That is the most important point. Now again, if you see how we have started with all the points that I have just now mentioned, that has been given in presentation. Now this was the drug under consideration, and we have worked on this particular molecule. Okay. So fingolimod, this was the drug which is used in the treatment of multiple sclerosis. Now, initially, we have prepared, we can say, sample of this particular drug based on the previous knowledge or the literature. We have prepared the mobile phase based on the history of that particular drug molecule in order to try, optimize the analytical method. Then lambda max has been selected based on their absorbers, and 218 has been considered as a lambda max for that particular drug. Now, initially, we have defined the analytical target. Now, since the drug is single, we have decided to work on the bulk drug. So, analytical target profile, we want to analyze that particular drug using HPLC. Okay. So, what target we can set as far as analytical method development is concerned? So, analytical target for any particular drug, it may be different for the different researchers. So, if suppose I'm using some different analytical tool like LCMS or LCMSMS, my analytical target may be different. If I am analyzing using UV visible spectrophotometry, the analytical target profile may be different. If I am working on HPLC for that particular drug, then analytical target may be different. So, in case of HPLC method development of fingolimod, we were expecting the robust method, which was again a base part of this particular QBD analytical method development. So, when we analyze any single component, USP tailing decides, we can say, one of the important parameters to judge how effective the method has been implemented. Number of theoretical plates, it is again another important parameter which will judge how efficiently the method has been developed. Then we can say retention time. So analysis time, it is also considered as one of the important parameter, and all these particular parameters can be considered as the best response. And we want to develop the analytical method with the USP tailing less than 1.2, number of theoretical plates as per the requirement, and short analysis time that is less than 10 minutes. So less than 10 minutes, it has not been written anywhere. But as far as industrial applications are concerned, that analysis time less than 10 minutes is considered as a best as far as analytical method development is concerned. Now, once we have decided this particular analytical target profile, then instrument qualification has been done as per the requirement at that time. But I am doubtful whether this particular qualification parameter will be introduced in ICH Q14 or not, because everybody, the instrument which the industry is using, they have been always been, we can say, qualified with reference to their installation. But as far as operational qualification is concerned, that operational qualification may be the mandatory part in for the ICH Q14 guidelines. So these are the parameters that help for initial, we can say, qualification of the instruments and the method. Then initial risk assessment. So what is risk assessment? It is the determinant for the effect of input variable on the process. From the risk assessment, one can recognize what are the critical attributes that are going to affect the final quality of the product. So Pareto analysis and fishbone diagram, they are the important, as we can say, tools for this risk assessment we can use. Now, initially, we have to develop a chromatographic condition. That initial chromatographic condition can be developed based on the history of the molecule, based on the, we can say, polarity of the molecule, based on some of the data which is available from the, we can say, clinical studies. Once you develop the initial chromatographic condition method, then that initial chromatographic method will be then subjected for the, we can say, QBD framework, and under QBD framework, the developed initial chromatographic conditions will be evaluated whether the initial developed method can be used, we can say, as a more robust method, or the method which we have chosen needs some modifications. Now, once we define the initial chromatographic conditions, and in our case, these are the initial chromatographic conditions. We are used this particular C8 column and the mobile phase methanol ammonium format with this particular ratio at 218 nanometer, flow rate 1.3 ml, and retention time was 5.15. Then we have initially designed some optimization models, and pH of that particular mobile phase we have changed using acetic acid and triethylamine. Now, as far as method optimization is concerned, method optimization we have done using response surface methodology by applying a 3-level box Baker design with the 3 center points. Basically, the 3 parameters we have directly selected here. So one more step is there where how we can decide which 3 parameters are important as far as the method performance is concerned. So large number of parameters are contributing, or that can be varied in analytical method development, including pH, flow rate, concentration of organic phase in the mobile phase. Besides this column on temperature, all these are the parameters that can be varied, and these parameters are always considered as you can say variables. And these parameters needs to be initially we can say segregated, and that we can say segregation or shortlisting of the parameters which are more critical. It can be done using Plackett-Berman design, which I have not shown here. And once you decide these three parameters are more important, so based on the results of that particular Plackett-Berman design, we have optimized this particular parameters, that is pH, flow rate, and methanol concentration. And based on the data generated and based on their we can say number of theoretical plates, USP tailing, we can say, no, not number of theoretical plates, USP tailing, we have decided what is the optimized condition. Now once the optimized condition is obtained, the optimized condition may be same as the initial chromatographic condition. It may be different than the initial chromatographic conditions. So that particular run you have to record, and you have to see whether that particular optimized condition is matching as per our requirements, which has been mentioned in the analytical target profile. So whatever that expectations we have given in the analytical target profile, whether these expectations we can achieve in using the optimized chromatographic conditions or not, that can be given, and that can be checked after optimization. Now, as I mentioned just now, validation remains a formality as far as analytical method development is concerned. So we can validate that analytical method development, but it is not our objective, or it was not our objective. So it is not a method intent, but it is an additional checkbox that will help to gain the additional confidence for any analyst whether that particular method which we have developed is really we can say having we can say applicability or not. So again, we have validated that particular method for the linearity, accuracy, precision, and robustness. We have done the robustness. We have developed the method under using robustness or exploring the robustness thoroughly. That's why all the parameters which we have evaluated as far as validation is concerned, it remains a formality. Now again, part of so these are some of the important results that we have shown, where this particular response surface diagram helps in effect of two parameters. By keeping any one of the parameter a constant, we can evaluate effect of two parameters on USP tailing, and based on these particular observations, we can determine what should be the optimized condition, and this is the optimized condition for that particular drug. And as I just now mentioned, that optimized condition you can run, and that optimized condition we can evaluate whether all the parameters that we have mentioned in the analytical target profile, they are we can achieve that all these parameters are not. Once we achieve these particular parameters, and we can say that this particular method has been developed using PBD framework. Now, practically explaining everything or explaining a small thing related to the analytical method development under QBD framework, it is not practically feasible within say 1 hour and 1.5 hours. So it requires say 2 to 3 hours for giving the detailed explanation how to initially screen the parameters using Plackett-Berman and how to go for the next, that is box bacon, for further optimization. So I have given just an overview. The basic objective of this particular lecture is to give an overview of quality by design and how we can effectively implement this quality by design for analytical method development, what information is available as far as regulatory requirements is concerned, and what which guidelines are expected, and what should be the content of guidelines which we are expecting this year based on the previous data which has been published. Now, as such, in conclusion, QBD, it involves understanding the process. Once you understood the process, the success of that particular process, it all depends on identifying the important steps which are, we can say, risky. Again, reducing the risk involved in the process and controlling that entire process, it ultimately leads to the successful process. So it is a regulatory requirement. So we cannot avoid that QBD for all segments of pharmaceutical development. So moving within the design space would not require any post-approval changes, which is the most important objective of any regulatory body, because they want to avoid the number of supplements. They want the initial submission, which should be with design space, so that any change, if any we can say inventor or manufacturer is doing within the design space, it no need to go for separate supplements for with that particular application. So US FDA has mandated QBD approach for the generic products from 2014. All the sectors have been covered as far as drug development process is concerned, except analytical method development. And into 2018, the concept paper has been published. They have given the timeline of 2018, 21 December, but because of the current pandemic, they have enhanced this particular timeline up to 2022 December. And these guidelines, with the hope that there will be more clear guidelines if you want to work in the area of analytical method development. So with this, I am going to conclude my lecture. So again, these are some of the you can say articles that we have published on quality by design. So this was initial review that we have published in say European Journal of Chemistry. So in 2014, where much information was not available on that area, and this is one of the article which is based on analytical method development under QBD framework that also we have published in 2014. But now uh, much information is available as far as how we can proceed with analytical method development in QBD environment. So we can proceed with we can say much expectations. And so this is the book which we have written on basics of quality by design in the pharmaceuticals. So again, I'm thankful to my respected guide, because of whom I'm in front of you. Uh, Principal of the institution, who Dr. Joshi sir, who has invited me for interacting with you to share my views on this analytical AQBD we can call. And I also am thankful to our in charge principal, Dr. Abu Bakar sir, who has given the permission to interact with you. So thank you.
Thank you, sir. Thank you so much. The session was very excellent and informative, and looking into chat box or participants has also benefited by the guidance they have got from you. While the presentation was going on, the participants were asking some questions regarding today's topic, as it is very important topic and important part of the research work. We would like to pick a few questions from them. Shall I proceed for the question answer session, sir?
Yes, 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 yes.
Yes, sir. Sir, first question is, what is the significance of quality by design in new method development?
See, any analytical method development, henceforth, till now, we are developing any new method as far as analytical method development is concerned. So, QBD has made mandatory for all types of analytical method developments henceforth, because they are slowly, we can say, covering the different segments of analytical pharmaceutical development. Now, analytical method development, they have not made it mandatory till date. But once the ICH guidelines will be there, that is Q14 guidelines, then we have to develop the analytical method, and we have to develop the analytical method as per the ICH Q14 in future. So that's why QBD right now there are no mandations as far as analytical method development in QBD framework is concerned. But maybe after 2013, everybody will ask you whether the analytical methods have been developed as per ICH Q14 or not, and that's why it is mandatory for all the analytical methods which will be developed in future.
Yes. So next question is, what are the quality by design approach for software or computer system validation?
Software and
Computer system validation.
okay. Now see, quality by design, it is a process dependent, we can say tool, we can say. So understanding the process, whatever may be the process, in case of pharmaceutical development, we need to understand the different processes involved in the pharmaceutical development. So whatever may be the area of science, as I mentioned, this quality by design tool, it is not, we can say, invented by the pharma people. It has been invented by the engineering people also. So they are using extensively for the different processes which are related to the chemical engineering. You can see the research papers which they have been published in 1970s, 1980s, 2000. Okay. So as far as software development is concerned, it is a process. So developing the software, we need to understand that the software development process, and you have to work accordingly. So the basic theme remains same. Whatever may be the process in science, we can implement this quality by design for that particular process. So whether it may be in the biotechnology, whether it may be in a software development in case of computers, whether it may be any of the extraction process, whether it may be those who are working on the extraction, we can use this particular quality by design tool where understanding that particular process, so extraction is a simple process. We can understand that process, we can change the composition of solvents which we have used and effect of that particular changes on our, we can say, output of that particular method. So as far as software development is concerned, it is an again a simple process. We need to understood the process, whatever maybe the process is.
So next question is, already we are using ICH guidelines for the method development. Then what is the need
Okay.
of quality by design?
Yes. So as I mentioned, since we are using ICH guidelines they have been framed in 2006, if I'm not wrong. Okay. These guidelines were initially developed for the primary analytical tools, which were very important at that time with respect to photochemical experiments. Now, they have been focused on initial development followed by the validation. So, in case of quality by design, the much focus has been given on only robustness. So, what is the difference is if you develop the analytical method and if you want to go for change in the method after development, ho sakta hai, if initially after development of the method cannot be applied for all types of, we can say, the formulations. If I have mentioned that this particular method can be useful for bulk drug, and if the formulation will be changed, then the method will be changed. So all such changes, if I am mentioning that all such changes cannot be covered by that particular method. In this situation, if you develop the method under QBD environment, then we are giving the design space and we are giving a very wide range where any change in that particular design space need not be mentioned as a change during the application. So we can proceed with the application with the design space and further applications of the supplements related to your original applications can be stopped. So that is the most important thing. Even if ICH guidelines have been given, they were very primary guidelines that has been framed in 2006, which was the need at that time. But the time has been changed. We are adopting the new science. The new science has been adopted to all sections of the drug development. And obviously, analytical development is an integral part of drug development, and it will be made mandatory, and that's why it is needed.
The next question is QBD is useful in molecular docking of new chemical entity?
Yes, QBD can be used in all sectors of science, as I mentioned. It is depending on the process, what software molecular docking is. We can use in all sectors. So, people have not used. Whenever there is a process where any process is divided into small segments, success of the process depends on how effectively you control the process. So molecular docking is a simple process. It involves large number of steps. If you control all these steps, ultimately we can work on, but nobody has worked it. Okay?
Yes, sir. So next question is, can you please explain about design space? Something about design space?
Design space, as I mentioned, it is a multi-dimensional combination of input variables. So design space, it is, we can say, large number of parameters that can be varied. Okay, all these parameters have been considered and the design space is given. Again, the composition of design space, it depends on what process you are studying. If I am working on the analytical method development, the design space will be different. I need to consider there the material attributes like the properties of the analyte. I need to consider the, we can say, the process or analytical procedure which I am following. So design space, it is a multi-dimensional combination of all the input variables. Input variables includes the properties of the material under consideration, the analytical tool which I am using. If I am going thinking about any small process which is related with the formulation and development, in that case, I have to consider the properties of drug, properties of excipients. Besides that, I have what methodology I am using for that particular formulation. All the process variables related to that methodology are considered in the design.
Sir, last one question is there.
Yes.
On which stage we have to apply the quality by design in our research project? means
At
on
which
which,
stage?
at
Yes,
which
yes, yes.
stage? Yes, sir.
Hey, uh, quality by design, this particular, we can say, technique, we cannot apply in midway. We have to plan your process through quality by design. So initially we have to define the objective. Means you have to start your process using quality by design from the start. You cannot, and we can say, use any we can say quality by design tool from the center point. So we have to start your process. You have to considering the quality by framework only. Means any of the mid stage we cannot apply quality by design.
Thank you, sir, and thank you all the participants. Now we will over our question answer session, and I would like to give a huge thanks to Dr. Sangshetty sir for sparing their valuable time and knowledge with all of us. I must say the session was so beneficial for all of us, and most of us will try to implement the things we have been highlighted in today's session. I would like to request Dr. Shivshankar Gunjegaonkar sir to present a vote of thanks to Dr. Sangshetty sir.
Yes, I'm audible to everyone. Okay. Good afternoon to one and all. Myself, Dr. Bunjaukar Shivshankar. It gives me an immense pleasure to express my vote of thanks to the today's renowned personality, Dr. Jaiprakash Sangashetty sir, Associate Professor at Y.B. Chavan College of Pharmacy, Aurangabad. Today we are so fortunate that to hear the Dr. Sangashetty sir. Sir has provided a significant insightful on the topic that is quality by design in the pharmaceuticals, specifically the analytical perspectives. Sir has given a detailed overview of the QBD approach for the analytical method development, how the QBD approach will help to get the predetermined specification during the development of the drug, understanding the process and parameter and how they are interrelated in order to the QBD to apply the QBD approach and its reproducibility. ICH guidelines, sir, has specifically given an insightful on the with regard to the concept paper, which is addressing the ICH guideline Q14. Sir, we are very thankful to the Dr. Sangashetty for enlightening us on this 1 week international e-faculty development program. Thank you, sir, for sharing your valuable time, knowledge, and the key aspects with all the participants. I'd like to express my sincere thanks to the management of Adarsh Shikshan Prasasat Mandal, the principal, Dr. Amol Joshi sir, KT Patil College of Pharmacy, the conveyor of this 1 week faculty development program, and the organizing team. Last but not the least, I would like to express my sincere thanks to all the participants who have enthusiastically participated in this particular 1 week faculty development program. Thank you one and all. Now I will hand over to the Ms. Chitra for the next process. Thank you, sir.
Thank you, sir. Thank you, Gunjegaonkar sir, for presenting vote of thanks. And thank you once again, Sangshetty sir. I am presenting this virtual certificate. Please accept it, sir. This is our small effort from our side.
Thank you. Thank you very much.
Thank you, sir. Thank you once again. Now, I would like to request this Gita Sapkai ma'am to start the valedictory session.
Thank you, Chitra. Welcome back. Check whether I'm audible or not.
Yes, ma'am, you are audible.
Yes. Myself, Geeta Sapkale, Assistant Professor, SP SIMS, KT College of Pharmacy, Usmanabad. Welcome back all the participants for the validity session. I'm just giving an overview of this 1 week International Faculty Development Program on Herbal Medicine Research and Global Health, a therapeutic perspective organized by ASPS, KT College of Pharmacy, Usmanabad from 9 to 14 February 2022 through StreamYard and our own YouTube live platform. The key success of this EIFDP was surely based on the presence of eminent and very renowned resource person we have got from national and international level. The EFDP was inaugurated on 9th February with gracious presence of Head Department of Chemical Technology UDCT and former Pro-VC Chancellor Dr. Babasaheb Ambedkar Marathwada University Aurangabad, Dr. Praveen Bakte sir. The first session was started with an excellent talk by Dr. Prashant Shamdula sir, Associate Professor from Government College of Pharmacy, Aurangabad. Sir has given a session on IPM in herbal medicine with their remarkable presentation. On second day, we have arranged the session of Dr. Uday Devre sir, Associate Professor, R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur, on potential role of nutraceutical as an immunity booster. On third day, we have an international speaker, Mr. Prathamesh Deshmukh sir, Senior Manager, Regulatory Affairs, Bristol Myers Squibb, New Jersey, USA, who had oriented us about regulatory aspect of herbal drugs. On fourth day, we have Senior Principal Scientist, Dr. Ashok Giri sir from CSIR, National Chemical Laboratory, Pune, who have briefed us about plant specialized metabolites in healthcare. On fifth day, we have Dr. Uday Devkote sir, Associate Professor, Government College of Pharmacy, Aurangabad. He has given an informative talk on role of HPTLC in identification and purity checking of herbal drugs. And today we have Dr. Jayaprakash Sangashetty sir, who have beautifully explained a concept of quality by design in pharmaceuticals. On behalf of Adarsh Institute and all faculty members, I would like to mention my heartfelt gratitude to all the resource person. It is very honored that we got these gems for our EIFDP. Thank you one and all dignitaries. Now, vote of thanks. I would like to give a huge thanks to all the knowledgeable and renowned speakers for sparing their valuable time and knowledge with all of us. I must say all the sessions were very informative and helpful for the upgradation of knowledge. We will be always grateful to all of them for their kind cooperation for success of these EIFTP. Thank you so much, respected dignitaries. I also would like to mention my special thanks to Principal and Convener of this EIFTP, Dr. Amol Joshi sir, for arranging such a nice program and supporting all us wholeheartedly. In fact, all I can say this, International Faculty Development Program have set a perfect benchmark by our institute in the rural area like Usmanabad in the field of herbal medicine research with a different perspective at national and international level. Thank you very much, sir, for keeping faith on us, gave us the kind support and appreciation that we always want from you. With gratitude, I wish my humble thanks to the management of Adarsh Institute, the backbone of Adarsh Shikshan Prasarak Mandal, Shri Sudhir Anna Patil sir, Administrative Officer Shri Aditya Patil sir, and all body members for endless and enormous support. Thank you one and all. I also would like to give big thanks to IQC Coordinator Mr. Pandey sir, Program Coordinator Mr. Alkunte sir and Chitra Madam, Organizing Committee members Umresh sir, Sharad Mani sir, Khan sir, Computer Operator Krishna Khomre sir, for all technical as well as non-stop efforts during the program. Without this support, it was impossible to conduct it smoothly. Thank you all. I also would like to give special thanks to all my senior faculty members, Dr. Aruna Devi Birazar ma'am, Dr. Amir Kazi sir, Dr. Tole ma'am, Dr. Gunjegavkar sir, and my junior as well as fellow colleagues and non-teaching staff for supporting us constantly to conduct the program. Thank you all. Last but not least, I would like to mention my special thanks and heartfelt thanks to all the participants for their overwhelming response for this EIFDP. Without your support, it was impossible to conduct the program. There were some technical difficulties occurred during the program. We really apologize for that, but you all kept patience and cooperated us. We hope we fulfilled the objective for what we have worked for and tried to give our best. With this due respect and on keynote, we would like to conclude our program. Thank you one and all. Now it's over to Chitra.
Thank you, ma'am. Thank you once again for the valedictory session and vote of thanks. And thank you once again for Sang Shakti sir for giving their valuable time to all of us. And they have given their valuable time and they have expressed their knowledge with all of us. Thank you once again. Thank you very much. Thank you one and all. Thank you all the participants. One note is there. Feedback, overall feedback link will be shared in the respective WhatsApp and Telegram group. You can fill that feedback form and the certificate will be received up to 20th February. You will all the participants will get the attending certificates. So have patience and have a good day. Thank you once again.